Thanks, and thanks to the organizers for having me here. It's it's a very fun group of people and a very nice set of topics. Um, the the stuff I'm going to talk about is fairly new work. It's a work that's maybe a bit off the central theme, at least the way I'm sort of presenting it right now. But hopefully, as we go along, you'll start to see connections between what I have here, the stuff from today, and the stuff that others are interested in. And as I go along, I'll also try to demystify what this title means and why I find it interesting, and what work we have done along this thing. A lot of the work that I'm going to talk about was done by this brilliant postdoc, Cal Floyd, in collaboration with Aaron and Arvind at Chicago. Okay, so I'll start by throwing up an input-output kind of relation that I find fascinating, and this is the kind of an input-output relation from a very, very, very simple gene circuit. So what's going on here is you have a caricature of a gene that's being expressed by that's being read out by an RNA polymerase, and it can do that either in the presence or in the absence of a transcription factor. And circuits of the sort we've all encountered and seen in many contexts. And if you, for example, treat the output of this gene circuit, which is the rate at which mRNA is produced, if you put that on the y-axis, and as inputs, if you, for example, think about the amount of transcription factor bound, you sort of use that as the knob you use to tune how much mRNA is produced, or the rate at which mRNA is produced. We all have seen input-output curves that look like this. As you tune more and more transcription factor in, you can increase the rate at which mRNA is produced by the circuit, and you can get input-output curves that sort of have the sigmoid-like shape. You can also get input-output curves that have more interesting humps and other features. So for some parameters, you get an input-output curve in which you not just have a simple sigmoid, but you have a sigmoid and then a hump on top. So we've all seen curves like this. You can sort of solve for things like this. It's fairly simple. A lot of this talk will be sort of structured around investigating or reinterpreting curves like this as computing devices. So instead of thinking of input-output, I'll start to think about these things as some minimalist form of computing. So what would computing look like in this caricature? If I just have a sigmoid, for example, I have a device that is able to distinguish a low input from a high input through some kind of a transition. If you have a sigmoid plus a bump and the bump goes back down, you have this minimal device that is able to separate this input profile, the concentrations of the RNA transcription factor, into 3, 4, or more regimes and regions. So this is a very primitive form of computing. We don't think about input-output curves in this kind of a way. But what I'll talk about, or the talk will be structured around this kind of an interpretation. And as we go along, I'll try to convince you that this kind of interpretation is actually very fruitful. It exposes limitations on what biology can do and what biology can't do, what kinds of circuit architectures allow systems to compute, and this notion of computing will also become concrete as I go along. But that's sort of what I'll spend. That's what I'll spend my time talking about. Okay, and so this is in one dimension. You can sort of draw the same kinds of curves in higher dimensions. So here, if I have inputs that are not just the concentration of the transcription factor, but I also have the concentration of the RNA polymerase as an extra input. I can have input 1, input 2, and the generalization of the questions I asked in the previous slide would look something like this. Can I program this circuit to have a certain kind of threshold in this 2D plane such that you have some response here and a different response here. And again, just to reiterate and re-emphasize what I'm going to discuss, or the main framework of this talk, I'll be interested in trying to understand what kinds of boundaries can be tuned by a biochemical network, by a signal transduction network. I call them decision boundaries because they separate two different kinds of outputs. How sharp can they be? And I'll clarify what this means as we go along, but for now, I'll just say how much can these networks classify. So for example, here, I just have two regimes, a regime with low mRNA and a regime with high mRNA rates. Can circuits like this tile this kind of space, tile higher dimensional space into multiple classes? Can they draw many boundaries wherein you can delineate different kinds of responses? That's the question I'm going to ask. And before I go ahead, questions of this sort, if you were to ask them with simple things like this, you could just solve the math for this. It's fairly easy to do, and you can sort of through an exploration of parameter space, you can basically identify and answer all of these things. But a lot of what we are confronted with in these biological systems is extremely high dimensions, as Sean talked about this morning. And in that case, we want to be able to say what kinds of classifiers these systems can actually operate as, how much can they classify, how well can they classify, and we want to try and see if we can extract some ground truths, some universal rules that go beyond these kinds of simplistic caricatures that actually work for a broader class of systems. Yeah, sure.
Other network is between that, or there's just one network?
Okay, so good. So that's a great question. So in this particular context, what I have is just one network, and this network takes inputs. The inputs can be concentrations of transcription factors, concentrations of polymerases, and then it produces an output. So I regard that as my computer. If I look at, for example, problems that look like immune recognition with multiple ligands, then you have computers that are hierarchical. There are many different inputs and outputs that get sequentially fed in. For now, we'll start off with simple things, and we'll go on to more complex things later on. Yeah. Okay. Great. So that's going to be the overall structure of the talk. The other question, the other comment I'll make before I move on is problems of this sort. If you view them through the language of classic machine learning architectures, simple perceptrons, multi-layer perceptrons, and more complex things, we sort of know what is needed to draw decision boundaries of different sorts. We sort of know that if you have a linear perceptron and nothing else, it can draw lines, can draw planes. Not does it go beyond that? And as you add nonlinearities, as you add more layers, you can get more and more complex responses. So all of that is really well known. To us, what was not well known is how do we view the kind of computation that implicitly happens in biology through a similar kind of a lens. How do we understand what's effective, what's not effective, what's the notion of depth, what's the notion of breadth? And so those are the kinds of themes and topics I'll try to get to as I go along. Yeah, Akshit.
Just to follow this question about boundaries thinking. So already in your previous slide, there was something about sigmoid and simple sharpness there.
Yeah.
We'll be thinking about the high dimension as a different kind of
So
sharpness.
for now, let's table that for a second. You're right. In this, it's easy to think about sharpness in the high dimensions. It's not so easy. So I'll come to that, but that's a good point to keep in mind as I go along. Okay. All right. So this is all very simple stuff. Just to sort of motivate this from a different perspective, I'll introduce a post-translational modification problem that actually got us interested in all of this. And then we discovered some nice rabbit holes, and we spent all our time thinking about computing and less time thinking about post-translational modifications. And so I'll talk about that first, and then come back to these kinds of setups. So the thing that got me started along this line was this very specific kind of a post-translational code that is called, that's composed of sugar. So this is a caricature of a protein. And what you see here is this protein is decorated with a bunch of sugars. This kind of a modification happens in the Golgi apparatus. Proteins get fed into the Golgi apparatus, and sugars of different lengths get polymerized. Sugars of different branches get polymerized, get added on to the protein, and then the resultant thing they create is colloquially referred to as a glycan code. I used this terminology once in a talk, and I got called a sugar enthusiast, which I'm not. But just warning if any of you want to reuse this terminology. And actually pick this up from these very nice papers, some by Mukund Tattai, some by Varki, and others by Madan Rao and co-workers. And so this sort of a code is used to mark cell fate. It's used to mark what the cells are going through, and this code is highly amorphous. It's basically a bunch of sugars, different lengths, different branches, and this carries information that is read out and used in many biological contexts, including in immune contexts. So this is a very nice experimental work that was done by folks at NCBS and ICER Pune. And what they did is, just to sort of illustrate that this code is actually interesting even in a very minimal setting, they took cells and treated to the particular chemical that disrupts the Golgi apparatus. This is an undisrupted Golgi apparatus, and as you go along here, you get completely fragmented Golgi apparatus. And what they did, after doing this, is they analyze these cells using by passing them through microfluidic chambers that had specific kinds of proteins. These proteins are called lectins. These lectins end up recognizing these sugars and binding to sugars. In biology, these lectins, these are two different kinds. They're called dectin 1, dectin 2, end up recognizing sugars, and they end up activating a bunch of downstream pathways. And this whole thing is proposed as a mechanism by which this code gets read out in biology. What they did here in a very minimal setting is they measured the binding affinities of these different states that are created by subjecting a cell to a membrane disruptor. They computed the binding affinities to two different kinds of lectins, WGA and CONA. The exact details are not super important. And what they found was that if one is given these binding affinities, the binding affinity of WGA and binding affinity of CONA to a particular image or to a particular ensemble, you can actually back construct just from these binding affinities. You can back estimate the actual state the cell is in. And this is a very nice, simple experiment to illustrate the fact that the kind of distributions that show up, the kind of distributions of these sugars that show up, can be used to encode something about cell fate, about cell state, because you can figure out how things are binding to them and then back compute or back estimate what kind of cell state the system, this membrane was originally in. Okay? And so this is just to sort of illustrate that this code actually has a meaning. There is some information that's going on over here. Okay, so just to summarize again, we have these sugars that are read out by these lectins, and they activate a bunch of downstream signal transduction pathways. The signal transduction pathways, you can sort of model them in one of, or you can understand them in one of 100 different ways. I have this caricature of just a simple phosphorylation cascade over here. All of this can be posed as a computing problem. And what ends up happening, or so sort of one minimal way to pose this as a computing problem, is to think about the binding of the amount that lectin 1 and lectin 2 bind as inputs to some signal transduction pathway, and these inputs produce these inputs. You can sort of plot what different kinds of cells do if you plot them, if you view them in this two-dimensional axis. And what you want your signal transduction network, this downstream thing to do is you want it to be able to draw lines of different sorts. You want it to be able to draw lines that cluster different colors differently. So what's actually the underlying data here is data from cells that were infected with different kinds of bacteria. This is the lectin binding of, if we looked at 2 different lectin binding affinities, and that's how we generated this data, and we colored this data. And what we want a signal transduction network to be able to do is we want it to be able to take data like this and somehow draw boundaries that have these curves that classify this blob into 1, 2, 3, 4, 5 different colors. So that's the problem that we're actually trying to pose. Okay, and we want all of this to happen within the constraints imposed by biology, within the constraints imposed by biochemistry. Okay, this question, how can class boundaries be drawn by signal transduction networks, is something that we found as a basal interesting problem, and I'll spend some more time on that. Yeah. Sorry. Yes.
So the bindings that you are thinking about, this is happening on the cell surface.
Yes.
And that is affecting the state of the Golgi inside the cell. That's what you were showing in the earlier one.
So these sugars are produced in the Golgi apparatus, and once they're produced, they encode the cell state. This is sort of the decoding process where the sugars are produced and they're being read out by the cell through a signal transduction pathway. And I'm posing this as a computational problem in which I somehow need this signal transduction pathway to be able to distinguish, to be able to sort of say that this blob is actually composed of 5 different subblobs, and I wanted to draw boundaries. So if I sort of contrast this with what I showed in the first slide, I had a very simple 1D setup where I just had a sigmoid curve and I had low and high. Akshit asked this question of what happens in higher dimensions, and this is just the slight generalization of it. I have this input, and I want the system to be able to draw out one boundary here, another boundary here, another boundary here, another boundary here. And I want to be able to ask, if you're given a signal transduction network and you want to do this process, what kind of crosstalk, what kind of connectivity do you need to be able to do this thing? Now, as posed, as I'm saying this right now, this problem is actually seems fairly trivial because you would assume that, yeah, drawing boundaries, I can do it. I can construct networks that actually do it. There's no real surprises here, and that's how we went into all of this, and we assumed that this would be a small step to overcome, and then we can go off and actually think about post-translational codes. But as we looked at this more carefully, we found some interesting surprises that sort of is going to be the bulk of the talk, and I'll get to that. Akshit, yeah.
I follow what the task is, but just to make sure, do the colored blobs, just what do they represent? Could you say that again?
So this is stuff this is ground truth that I know that this thing doesn't know. What we did is we looked at published data sets where you can actually go and get the glycan mass spec data from different kinds of experiments. So this is an experiment where this is glycan data from cells infected with different kinds of bacteria, and that's how we've colored it. So if I gave you the full mass spec, you would see this big blob that's not colored, and then we went and we sort of identified things and we colored the blobs in this manner.
Okay, thanks.
Okay, yeah.
I think this is a projection.
Yeah.
Of some data in a higher dimension.
Yes. Yes.
Then in higher dimension, I could get away by just doing it.
Yeah.
So how is this problem?
So great. So the question is, if all of this is being actually read out, if all of this is being read out by all the lectins you have access to, then you can maybe get away with drawing simple kinds of things. We'll also get to that in a second. We'll also get to the high dimensional version of this. The low dimensional thing is more meaningful, at least in this sort of context, because in general, we wanted to ask what happens if you are constrained and you have access to a smaller alphabet of lectins. So in again the sort of published data sets that we have seen, the diversity of you do have access to a diversity of lectins, but the question almost always is how much can you get away or how much can you extract from a low dimensional projection? Because that the implication seems to be that that's sort of what's happening, right? But if you had access to everything, then you can really parse this out in high dimensions, and you don't have. It's an easier problem, but I'll also comment on that as we go along. Okay, yeah. Oh, there's one black line that didn't show up. I don't know why. So there's one more here. There should be, didn't show up. It shows on my computer. I don't know why it doesn't show up here. There should be one more black line. But that's really good. It's like 250 and you're really paying attention. That's super happy. Okay. All right. So just to sort of maybe drive home this point a bit more, this is a different kind of assortment. This is, if you do this, if you just look at glycan, again, mass spec glycan data, you pick lectins that you think are important, and you plot it in this sort of low dimensional space. Again, you see this, you see distributions of this sort for different stages of prostate cancer, and it turns out all of the blue blobs are actually the no cancer state, and all of these are the states with different kinds of different stages of cancer. And so if you want to distinguish between the blue and this colored mess over here, it's easy to draw a plane. But if you want to go in and try to understand how you parse out everything that's going on here, that's sort of a tougher problem. So there is a few different hierarchies that you can get into here. And this is the same thing that I showed you before, but plotted in a different way. All of this is just to sort of show that there is a nice computational problem you can sort of pose. We think this problem is relevant for how biology deals with it, but for now, let's assume that I'm posing it in this way, and we're going to try and understand how to draw boundaries. Okay, and so in all of these things, we're going to use we're going to try and see how one can or what kinds of architectures one would need to draw boundaries of the sort. But before getting into the signal transduction networks and doing all of those things, let's sort of make this problem even simpler to ask, to see if there is anything universal hiding behind all of this. And so before I go ahead, sorry, the one last thing I wanted to say was there is an enormous diversity in this glycan distribution, and the diversity also changes depending on the organism type. So it turns out if you look at lower level organisms and you take the mass spec data from glycan distributions, and this is work from Madan's paper, I forgot the reference here. If you take mass spec data from low dimensional organisms, you can fit them with the low dimensional Gaussian mixture model. This is just 3 modes. And as you go to humans, you sort of need much higher. You need a 20-mode Gaussian mixture model to fit things. And so there's information that sort of increases in complexity as you go through organisms. And so the spherical cow question that I'll sort of spend my time thinking about in this talk, and I'll actually make this an even more spherical cow as I go along, is if I have access to a small number of lectins, a small number of inputs, is it possible to tile space and classify and sort of extract something on the order of 10 to the power of m classes, where m can be on the order of 2 or 3? Okay, so I'll try to get to that, but I'll start off with something much simpler than even the spherical cow question. But before I do that, I wanted to just remind you that if one is interested in doing all of this, drawing class boundaries, and you had access to perceptrons, just any simple textbook, undergrad, high school machine learning thing nowadays, you can solve these problems very easily. But writing down analogous statements for biological networks, where concentrations, where activations are the equivalent of your weights, is slightly more interesting, and that's why we think this problem is worthwhile thinking about or working on. Okay, so that's what we'll talk about. Okay, so I'll start off by constructing with a highly simplified caricature in which I'm going to assume that my computing device, the device that sits underneath, gets inputs and does the computing, is a very simple chemical reaction network in terms of the order of the chemical reactions. So I'm going to assume that you have a bunch of first order chemical reactions effectively. So each node here denotes, for example, a molecular state. It could denote, for example, a configuration. And as you go from one node to a different node, imagine you have a first order reaction that's sort of taking place as you go from this to this. Okay, so that's the caricature that I'm going to have in mind as I think and talk about these things. And if I expand this caricature out, I can eventually go to higher order reactions. But I'm going to start with very simple things, just first order reactions, and I'm going to try and construct a computational engine with this. Okay, I'm going to assume that the reaction rates between to go between any two states is parameterized in this very minimalist manner. Each reaction rate depends on an energy that sits on a node, that it depends on a barrier height that separates two nodes, and it also has these extra components which we call affinities, and these are anything that's antisymmetric. So if you have, for example, a proton motive force, electron motive force, unbalanced pH, unbalanced concentration gradients, all of those things go into this extra affinity-like term. Okay, so this is the most minimal characterization, parameterization I can have for rates. It has everything that needs to be symmetric. It has all the possible ways in which you can have antisymmetry in this setup. Okay, so with this in mind, I'm going to construct a computer. I'm going to build a computer in the following way. I'm going to assume that I feed into this computer values of these f's along two of the edges as inputs. Okay, so that's what I'm feeding in. Think of this as being equivalent to these lectin activations you saw a while ago. That's what I'm feeding in. And the outputs are going to be the concentrations or the probabilities, depending on the language you're most interested in, of two nodes you have chosen. And in playing this game, we can play this game with arbitrarily complex topology. We can play this game with networks of arbitrary size. You can sort of play the game in any way you want. But this is how we are going to set up this basic problem. Okay, and so the question you're going to ask is, can we construct or train or learn a network such that any problem you give me, any sort of decision boundary you give me, is plausible. So what would that look like? So you might come to me and say, Suri, I want to construct this sort of chemical computer that takes as input values of these f that live on two edges. So these are two physical rate constants that I'm tuning. And outputs are two concentrations that I have to get. And I want it to be such that if Fa and Fb are in this region, I get the blue product or the blue protein. And if Fa and Fb are in this region, I get the red protein or the red product. Okay, and that's what I want. And then you can say, I want this to be sharp. I want this boundary to be loose. All of those questions are things we can sort of talk about. You can also add squiggles to these boundaries. You can make this boundary more or less complex. Okay,
that it's the F's that are input as a
no, it's not.
That's just as an example.
Yeah, we're starting off with simple things. I'm jet lagged, so we're starting off with really simple things. Yeah, usually I don't talk so slowly. Usually I talk much faster. But given the time zone I'm still in right now, I'm slowing down, which is a good thing for me if I want to slow down ever. So, okay, so how do I do this? There are a variety of techniques you can use to sort of train this. I'm not going to get into that. It's an interesting problem if folks want to chat about. But we give this network different kind. We show this network data of different sorts, and then we train it, and we sort of try and test the network and see if this works out. Okay. So what we'll do now is we'll sort of play a small game. Some of you in the room, Gautam, for example, has seen this game before, but others haven't. But we'll sort of ask if this kind of a computer can work or not work. Okay? So let's say I start off with one of the simplest problems, which is I have Fa and Fb, and I want to construct a Markov state computer such that blue is down here. I'm partially colorblind. I think this is orange, but I can't be sure. I think this is orange. Let's call it orange. Blue is down here and orange is up here. So any guesses for whether this kind of a computer is possible in what I told you? Yes. Someone said yes. That's like, it's obvious. It's a line. It's a straight line. If it can't do this, we should not talk much. It works. You can sort of do this kind of thing. What about something like this? I have 2 point clouds. I want to be able to separate the point clouds. Can you get a computer that, can you construct a Markov computer of this sort that can separate these 2 point clouds? Someone said no? Yes, no, shout it out, be confident. Yes, good, loud yeses, good. So you can find solutions that sort of get you this kind of thing. This is one particular solution. This is all numerical. We'll get to theory soon. This is all just numerical exploration. What about this problem? The same thing will happen. I'm just boring all of you right now. It's just the same thing. It's just different kind of solutions. I've just literally moved this thing around. Okay. Okay. Now let's just play the same game 3 more times, and then I promise you will be done. What about this thing here, the square? Great. Yeah, it's like almost similar. What about this pizza wedge? Yeah, right. I mean, it's a square. It's a pizza wedge. We are taught to respect or worship rotational symmetries, and so you think you do this, you can do this, and this is where the first surprise comes. This starts to fail. There's a very interesting failure mode wherein you find that the square works. This pizza wedge-like thing stops working, and there's a lot of failure points over here. Okay, this failure was interesting to us because we would have, we assumed that if you had the ability to tune the topology of this network, make it as complex as you want, make it as big as you want, you can effectively encode an arbitrary amount of nonlinearity in it. And so we assumed that the computational capacity that is encoded in these concentrations that you output should be infinite. You can do anything you want as you make the network larger and larger, more and more loops, whatever kind of thing you want. But this damn thing always fails. This is failing in this numerical experiment. I'll show you in a second that it always fails. It's a law of nature. It's not just an accident. What about this here? No, right? I mean, this is not going to work. And this is one of the simplest problems that biology should be able to solve. This is recognizing in from out. This is saying I have one thing here, one kind of response here. Everything is not that. And this is a very simple logic problem that biology should be able to solve. But you try solving it in this manner, and you'll find, as we found, that it can never work. So this was an interesting surprise. It sort of pointed out that when you think about posing these questions of expressivity of computation, a lot of things that you might have, or at least we assumed for granted, start to fail. And the reasons for this failure were somewhat surprising, and it turns out universal, and that's what I'll spend some time elaborating in the next few slides. And there are very interesting ways in which this failure can be avoided, which points to biological mechanisms that we haven't fully tested. We have some guesses, and that's what I'll spend the remaining, I don't know, the remaining minutes I have in this talk. Okay, all right. So all of this is great. I showed you these kinds of sketches where this was glycomic data for cancer and no cancer. And if all you want to do is draw lines, if you want to draw a simple decision boundary, just a line, you can do it with these kinds of circuits. You can do all of those things. But if you want to do something more complicated, if you want to distinguish between all these classes that are hidden in here, what this seems to suggest is you need something more than what I'm showing you right now. You can even if you take the most elaborate chemical reaction networks and you ask these questions as posed, as written down, they can't solve this problem. And so you need something more. You need something more interesting to sort of get to that. So that motivated for us the following kinds of questions. What sets limits on expressivity? So what's expressivity? This is a new word I've introduced. It's the ability to draw different kinds of boundaries. It's the ability to draw different kinds of shapes or different sorts. So remember in the first slide, I showed you this input-output curve. Input-output curve could be going up, coming down. All of that, we sort of think as expressivity. We sort of view that as the ability of a biological circuit to do something interesting in this sort of computational lens. What we found was that there's a fundamental limit on how expressive we can be due to a constraint that my postdoc, Cal, actually derived. And the constraint is expressed in a very, very simple manner. So imagine you pick up any StatMech, or not imagine, you all have picked up StatMech textbooks, so just go back and revisit that memory. So if you go back to basic intro StatMech or intro thermo, we talk a lot about responses. We talk a lot about how the properties of a system change when you start to tune different kinds of driving forces. And we know that these kinds of responses close to equilibrium are very well constrained, very well curtailed. They're curtailed by the laws of linear response. We know also that if you're in the thermodynamic limit, you have tight constraints on the signs of second derivatives. You know all of those things. We know all of those things. But if you go to systems that are really far from equilibrium, of the sorts that we are thinking about here, with affinities and all of these things floating around, in general, you can't say much. But what we found is that there is, interestingly, a very nice constraint on this quantity. So this is the rate at which the probability sitting at node K changes as you change the driving force along reaction Fij. Okay, and this could be any K, any Fij. It could be K sitting next to an Ij, K really far from an Ij. So construct this partial derivative. And what we found is that this partial derivative has a fixed sign across the entire range of Fij. If it is positive for one value of Fij, it's always positive. If it's negative, it's always negative. And we can prove this. I can go into this for folks who are interested later on the board. But we can prove this thing for any topology. And what this tells you is that at a fundamental level, the response that you can have in these kinds of non-equilibrium circuits, signal transduction networks, what have you, in this caricature, is dramatically simple, is much more simple than what you would have gone in expecting. In particular, because the derivative has a fixed sign, the only kind of responses that are allowed are either monotonic up or monotonic down and nothing else. You can't get wiggles, you can't get squiggles, you can't get any of those things. So in one dimension, that's all you can get. Monotonic up or monotonic down, and you're limited by that. There are also additional symmetries that pop out once if you stare at this curve. So this is a curve in which we have plotted the same quantity for different nodes. So we are tracking one Fij, and we are plotting this curve for different nodes K. And what you see very interestingly is that all of these curves go up and they have a maxima at the same, or maxima or minima at the same point. And what this tells you, and I'll come to this in a few minutes, is that you not only have this kind of a constraint, you have an additional constraint that any average non-equilibrium property, so you can construct any average property in the system, not just an average concentration, not just the concentration or probability of one species, but any average probability, averaged over everything in the system, and it turns out even that will have a fixed sign because of this sort of symmetry that you see here. So we can prove all of these things. We think it's a new kind of a non-equilibrium response relation that was just hiding in the algebra, if you will, that people didn't notice, and we can sort of see that. So what does this have to do with expressivity? You already can see maybe hints of an answer in one dimension. The curves that you get by doing this kind of a thing are constrained. They can only go up or come down, nothing else. If you play the same game in two dimensions, you can start to see that you actually have tight constraints on the kinds of decision boundaries you can draw. You can prove this in 100 different ways, but the easiest way to think about this is, imagine you want to draw a decision boundary that starts to bend around, that starts to bend back around at some point. Right around here, you have to violate this partial derivative constraint. You can't get away with that. And so the only kind of curves you can draw in this kind of a caricature are a bunch of quarter circles. That's the best you can do. You can invert the quarter circles. You can go this way if you want, but you can't. Once you fix one way of going, that's it. You can't get out of that loop. So all of this is telling us that there is that the computational capacity of these systems is fundamentally limited. You can draw curves, but the curves are constrained, and they're constrained in this manner. So this is both good news and bad news. It's bad because you would expect biology to be able to do things that are more than this, but it's good because maybe there's a way in which you can systematically overcome this constraint, just like in a neural network, or just like going from shallow networks to deeper networks. You could get more and more interesting kinds of decision boundaries. Maybe there's a way to do that in this kind of context. And indeed there is, and the solution is again embarrassingly simple, but seems to be universal. What we in the caricature that I introduced, I had a bunch of reactions. I had these Fs that lived in each reaction. If you think about the common kinds of things you end up tuning, if, for example, you tune pHs, you tune ligand concentrations, you tune the concentrations of different kind of binding agents, almost always these kinds of things that you tune affect not just one, but they affect a multiplicity of reactions. If, for example, you're thinking of a proton motive force that supplies protons or deficit of electrons for you, it doesn't affect just one reaction, it affects a multiplicity of reactions. And so that feature, the fact that anything that you tune is typically multiplied a bunch of times, when you think about this more mechanistically, is a feature that helps rescue systematically all these limits on expressivity. Another way of viewing it is just like in Andrea's talk, there's this thing of in the immune context, you have crosstalk between different kinds of receptors and ligands. That kind of crosstalk we call promiscuity. That's also a version of multiplicity. We think that systematically rescues you. So how does it look like? Imagine now we go back to this problem that ended up failing. But if I now feed this F, not just into one input, but into two inputs, just increase the complexity in that simple way, you can start to see that what was not possible starts to become possible. You can start to draw curves if you allow for the fact that each input, each F affinity that you're given is affecting two reactions and not just one reaction. Solutions of this sort start to become possible where they were not before. We can do this more systematically, but before I do that, all this is saying is, if you have promiscuity, if you have enzymes talking to multiple different things, if you have things like kinases in which you have one agent affecting multiple pathways, all of those are knobs by which you can tune how expressive that cascade can be and how it can turn around and do more interesting things than what you would have imagined. Yeah, Krishna.
If I redraw my input-output map as the 4 dimensions where the input actually change,
Yeah.
and then the output as the actual output change.
Yeah.
Now, because I'm just curious, like the same constraints we derived previously, what to do in that space?
Yeah.
It's just the projection.
Yes, exactly. So if you take the 4-dimensional space, and this again gets to you what you said, if you take the 4-dimensional space and they're unconstrained, you'll have all of these things. Then take a cut through that space, you start losing all those constraints, right? And the cut is important. The cut is a constraint that is biologically put in, because you're saying that this agent you're supplying is actually affecting two reactions, and it's a constraint you can't actually overcome. Okay, so we can do this thing a bit more rigorously than just simple numerics. You can go back to this quantity, this partial derivative, and you can try to imagine computing the number of times the derivative can change sign as this affinity is tuned in one dimension. This is a one-dimensional input. And what you can prove after a few lines of algebra, and it's a nice proof for anyone who's interested, is that the number of turning points, the number of times this thing turns back around, is bounded by 2m minus 1, where m is the multiplicity. It's the number of times the input you're providing is affecting reactions. The number of reactions the input you're providing, it's the number of reactions your input is affecting. That's this m, and that sort of sets the number of turning points. So if m is if m is 1, this sort of tells you that the turning points, you can have at most 1, but we can actually prove a stronger one, that's 0. And as you go higher and higher, 2, 3, 4, we have gone up to 5, I think. You can actually verify this. You can actually numerically see that this bound is almost always saturated. And what this means in practice is if you have m of 1, you can only do monotonic things. If you have m of 2, you can start having humps. And this kind of a thing in 2 dimensions is going to start looking like circles for you right now. Okay, and this humps is also there's also more interesting things here, because this is just allowing you to distinguish between a low input and a high input. This hump-like structure can actually allow you to do what looks like a band-pass filter, because you can distinguish region 1, region 2, and region 3. So you can start to systematically make these networks operate in more and more complex ways by simply tuning how the inputs you are allowed to tune affects the chemistry that is sitting inside the networks. And that's a very simple knob that you can actually tune to do this thing. Okay, all of this is in low dimensions. What about high dimensions? This is a weaker bound, but it still works. So if you want to play the same game in high dimensions, you can go back and look at how people in studying classifiers on high dimensions do all of these things. So there's a very nice concept called the VC dimension, which enumerates or which sort of bounds the ability of your classifier to separate out points with different kinds of complexity. So if you just have 3 points, for example, plus and minus, if you just have 3 points, and the game you're playing is I want my classifier to be able to classify all binary combinations of these 3 points. So in other words, there's 3 points, any combination of plus and minus, 1 plus 2 minus, 2 plus is 1 minus, all 3 minus or all 3 plus. If you want your object to be able to classify all of those combinations, a simple line suffices. That's all you need. So the VC dimension of a line is 3. If you go to 4, a line can't do it. You need a curve. You need a squiggly curve to do it. Okay, and so we can, for example, compute or at least try and bound the VC dimension of this chemical reaction network. And what we can show is that it grows exponentially with the dimensionality, but most importantly, it grows exponentially with M, this input multiplicity. So as you have one input going into multiple channels, you get computing power that is exponentially increasing in the dimensionality that you're actually measuring. And so this we think is it's still a very weak bound. We think there's a stronger bound lurking in here, but at least this sort of gives you this sense of there being something that's exponential as you increase the multiplicity. Okay, all of this is for 2 classes. That's the sort of the most boring version of this game. I told you about this glycan code. I told you about how you can have all these multiple classes. So what if you want to do that? What if you want to do multiple class classification simultaneously? Something like one-hot encoding. Can these networks actually get to that? And what we find is that similar kinds of constraints end up working out or end up existing. So if you want to simultaneously classify this, 4 classes with a 2-dimensional input, and you are constraining yourself to use Markov networks or chemical reaction networks with no multiplicity, you can't do it. You can never classify. But as you start to, and we can actually prove that this is 3, you can't go beyond 3, but as you start to add in multiplicity, you can start to play these classification games, and you can start to do better and better. Yeah. Yeah.
Now you're saying at least you can draw one boundary in FA.
Yes.
One boundary in FB.
Yes.
M equals 1.
Yes.
That gives you high, high, low, low.
Yes, but I want to simultaneously draw all 4. So I want to say this is green, this is blue, this is red, and this is orange. So I want to simultaneously draw all 4. So in other words, the game that I'm playing is slightly different, and I'm saying, and this will become clearer when I connect it back to something like one-hot encoding. So what I want is, I want this network to do one-hot encoding, and I want it to be able to find solutions that do one-hot encoding. And what I'm saying is that if you want to do one-hot encoding with 4, you can't with just M of 1. You need M of higher than 1, but we'll sort of generalize this a bit more as we go along. But the construct you had in mind is perfect. It's okay. I'm sort of imposing a very specific problem when I sort of do this kind of thing. Okay. Okay, so all of that works out. So in the remaining few minutes, I'll talk about a few things that a few results we found in literature that sort of are in line with the kind of things we have been finding, or our findings help support what's been observed in literature. We're still working on generalizing this to the glycan code. I'm happy to chat more with you offline about where we are in that process. But these are our first preliminary indicators that these kinds of ideas may actually be useful or may have been lurking in the background for all this while. So as I told you, if I just have one input and I want to create something that's not a sigmoid that has a hump, you need a multiplicity of 2. There's this very nice paper that came out fairly recently in Nature Chem Bio, where they wanted to construct they wanted a setup where where they wanted to create a band pass filter with a protein, and the minimal thing that they had to do in order to enable this is they had to enable this ligand, this drug, to actually bind the protein in 2 different ways. And if you do that, you sort of start to get these band pass filters, and that's sort of what we would predict in our minimalist caricature. But this is something that you can also use as a design problem. So everything I talked about so far, I said it's effectively for first order reactions, and a lot of what happens in biology is higher order reactions. It's groups of reactions coming together with different timescales. So can all these ideas work if you have higher order reactions? And we think we can show that it can in a minimal setting, and we are generalizing this right now. So imagine you wanted to do a computation with a 3-node computer with a 3 with a computer that has three compounds, A, B, C, and you also have this additional bimolecular reaction that almost looks like catalysis, or you have this bimolecular conversion like reaction. Even in these two contexts, even if I just tune the input along one of these reactions, the fact that it's bimolecular, we can show automatically induces a higher order multiplicity. It automatically allows the system to start having curves, and you can get profiles that go up and come down, even though you just have one input. So if you have nonlinearities in your reaction, they can interact cooperatively with the inputs that I'm showing here, and you can build up a higher order multiplicity implicitly in that way, and you can sort of get better and better computational abilities. This sort of goes really well with recent work from many groups, Sergei Maslov, Alexei Elovitz and co-workers, where they tried to use a dimerization network or a system of dimerization reactions to construct chemical computers. And what they found is that this kind of a dimerization network can actually do a lot of interesting classification tasks. And that would be in line with the kinds of things we are seeing. These kinds of networks should be able to classify. They can go up and come down. We haven't exactly computed bounds or limits on things like this yet, but it sort of is in line with the kinds of things I just that show up here. And this is something that I found super exciting. This is work that this is actually a nice paper from Gasper Tachik, Bill Bialek, and co-workers, where they looked at this old problem of how a biochemical circuit can take input morphogen gradients and get output gene patterns. And in this paper, they sort of just did a brute force search and tried to find architectures that can take these morphogen gradients and get you the kinds of gap gene patterns that you see in experiments. And what they noticed as they were sort of doing this brute force search is you needed these morphogen gradients to talk to a multiplicity of reactions. And that is a minimal ingredient that was necessary in order to go from here to here. If you didn't do that, you couldn't take these morphogen gradients and transcribe them into these gap gene patterns. And that's in line with the kind of stuff that we have said, but viewed through an information theoretic lens. So let's say I repose our problems in terms of maximizing mutual information. What we can show is that if you have a higher input multiplicity of 2, for example, then you can send an input in, and the input looks like this dashed line with these 3 peaks. And if you have M of 2, the system can tune the concentrations of different states of different chemicals in your reaction network such that it effectively one-hot encodes this input into the probabilities in your reaction network. So in this particular case, what happens is the first peak you have one kind of product, the second peak you have a different product, and the third peak you have a different product. So all of this happens automatically. It doesn't happen if you have low input multiplicity, but for high input multiplicity, you can start to get one-hot encoding. You can extract all the mutual information you want to get from things like this. I have a very small amount of time left, I think 4 minutes or something of that sort. So I'll not spend too much time on the last part. So this is just one last thing. This is the glycomic stuff. We can sort of draw lines. But I'll spend a small amount of time on a related conceptual question that we found super interesting, which is the question of control. So the previous one was about computation. This is now about trying to figure out what you need if you want if you have the situation where you have a signal in the environment that's changing as a function of time, and you want your system to be able to track that signal and reproduce it, hopefully, in a manner that's as faithful as possible. Okay? So if you want to solve problems like this, the question we got interested in was what kinds of constraints are needed on biochemical circuits in order to get them to solve problems that look like this. And what we found was that as a consequence of this partial derivative constraint that we found, problems of this sort can be solved fairly easily without any specific constraints on architecture, on biological architecture, on network connectivity, so on and so forth. So how does that solution actually look like? Because we end up having this constraint on the partial derivative, let's say I want my chemical reaction network to track some environmental signal that's sort of fluctuating up and down, and the fluctuations are not too rapid. That's the one sort of constraint we need. What our results tell us is that as long as we have a linear integral feedback in which the environment is compared to the state of the signal, and the rates along any chemical reaction are updated in this very simple manner, we can then guarantee, because we have this constraint, we can guarantee that this time-dependent system, which is fluctuating, it's changing as a function of time, this time-dependent system will end up tracking the environmental signal you want to track. Rules like this, we think, can actually show up very naturally through a variety of means. So in this particular thing here, I can tune, for example, the amount of transcription factor, the rate at which the transcription factor is changing through the stoichiometric rule, which is very easily implementable, that implements stuff like this. And I can get the gene product, the mRNA here, to track what's happening in the environment. As a consequence of this constraint, this kind of a feedback rule on any chemical, on any edge, allows me to control the concentrations on any node through a very simple mechanism without any fine tuning. What's super interesting for us is that even though what I've told you is just in one dimension, you can actually generalize this to higher dimensions. I have this plot here where I have now, I'm trying to track a vector signal, not just a simple scalar signal. What this plot shows, I'm not going to go into this in the interest of time, is these kinds of rules exist even in higher dimensions. And if you want to solve feedback and control problems because of constraints like this, all you need are very simple integral feedback rules, and you can solve control problems. You can also solve a lot of adaptation problems with this thing without having specific kind of modules, specific kind of requirements on the architecture. So I'll stop here. All of this work was done by this postdoc, Cal, in collaboration with Arvind and Erin. Thank you for your time, and I'll take questions anymore, if any. Thanks.
Hello, does this work? Okay, great. So I wanted to get a little more of a sense of why there's interest in creating these sort of chemical ways of doing computation. Is it like a model for how life, basically posing life as a computation problem, or is it more like, do you at some point foresee using them for actual computations like we do for like, our current computers, but maybe for like different kinds of problems, perhaps?
For me, it's mainly the former. It's not the latter. It's a way for me to sort of there's this standard systems biology narrative of input-output, of tuning things in a particular manner. There's the complementary dynamical systems perspective that tells you get all of these kinds of, you get these regimes. This is, I think, a different axis that is productive to look at. Now, this question of computing with the question of actual chemical computers, currently no, but there are themes that there are concepts like reservoir computing, for example, that show how you can use these kinds of things as the engine for a general purpose computer without actual without gates and without any of those things. And so maybe in the future, yes, but everything here, for me, the motivation was the former.
That we can have the classification maps, right? So is there a way we can do the reverse? Like we know a lot about what kind of classification map we have. Can we create some kind of complexity in the network from that data? Not really, right?
So a bit, yes. So for example, if you have a system like this, it's able to draw X kind of class boundaries with some complexity. We know that there has to be through ideas like this some amount of crosstalk. And so that's the kind of prediction we can make at this point. It's minimalist. But if you take these ideas and you more tightly constrain them to work with existing signal transduction networks, then you can make more. You can make predictions that are more relevant experimentally. But for now, what you can say is there is a limit, and if you see that you're able to do something, it tells you the very minimal that there is crosstalk in your chemistry under the hood.
Okay, thank you.
Okay. I maybe had a small last question about the gap gene networks, because it seemed to me that what Thomas and Gaspar and others, like the fact that these inputs talk to multiple gap genes and then you get more information, that makes sense. But I thought you were maybe saying something stronger, which is that if I have a single input or a uniformly impressed input and it talks to two different rates, then I can create more information, which is slightly.
So sorry. Yes, I was saying. Let me just sort of be very precise in what I was saying. I was saying. So in what they did, they said, yeah, if you have these genes, you need these to. These are inputs, and you need these to talk to each of these. You need to talk to multiple reactions, right?
But you're saying more almost. You're saying because these inputs all have shapes, right?
Yeah.
But you're saying in a way, or the previous thing with the, I think on the previous slide you had an example where you had presumably what could be like a uniform additional input, and that can talk to the rates, right? Here they don't talk to the.
Oh, sure, yeah. So in principle, I could shape my Fs in different ways. So for example, I could send an input to one edge that is the F, a different edge. I could send an input that's not the same F, but it's a differently shaped F, and the same kinds of things would happen. So you would get. You would be able to extract more mutual information. You'd be able to do one-hot encoding and all of those things from all of that. Yeah.
We still have time. We spoke about expressivity, shapes we can draw. It's also the concept of versatility or flexibility. So once I build a complex network, can I easily fine-tune it to solve other problems?
Yeah. So this comes to this question of how much free space, how much free breathing space you have. So I had these bounds at some point. Blah blah blah. So I had these bounds at some point. So let's say we have this problem where we need, for example, let's say we need 1, 2, 3 turning points. Okay. And if we do that with an M of 2, for example, we have very little leeway in terms of what we want to do. But if we do the same kind of thing with an M of 5 or an M of 6, we'll start to have more. We'll start to have the ability to sort of tune things more flexibly. So that's the sort of easy answer I have. We haven't thought more deeply about what you said about how flexible you can be, but I think the answer is you probably want to be way below what you are allowed. And if you do that, then you can do this thing. The other analog of this question would be, what if you had to tune these things on some evolutionary time scale? What's the best thing? My guess, which we haven't sort of contrasted with any kind of experimental data so far, is you would probably want to have a lot of gap between how much you're allowed and where you are, so that you have the ability to undergo further fine tuning. So those are my 2 short answers to that.
Do you mind asking after? Okay.
In one of the cases that the capability.
No, go ahead.
Oh.
No, go ahead.
Yeah, go.
All of those are symmetric about 0,0 kind of. So, I'm thinking that it is coming from the fact that your derivatives have kind of symmetry, so you can classify this symmetric picture. Would it happen that if your circle was centered around not 0, 0, but somewhere else, it still be able to classify those?
Yeah. So the basic underlying theme will still hold. So if you move the circle around, for example, you may find that the details are a bit different, but you will still find that you are limited in what kinds of shapes you can draw if you have M of 1. And if you have higher value of M and you have a circle that's not at 0, that's sort of very far away, it'll be able to do it. You just need more degrees of freedom. So like the degree of freedom that comes with this input multiplicity is interesting. It allows you to bend around in different ways. Moving around the origin, I don't think that affects it. It's going to sort of have the same kind of picture. No, so I can sort of reparameterize things very easily, for example, and that will sort of change that, and that will take care of what you're saying, right? Okay.
Then
Okay.
I would say let's thank Suri.
Thanks again.